Now that we had seen what happens to glucose in the end of glycolysis, we had obtained two molecules of pyruvic acid. Now we are going to see what happens to that pyruvic acid. Now what intrigues me a little bit is that we started in the beginning that respiration is the process in which the glucose molecule would break down into 6 carbon dioxide molecules and 6 water molecules. And what we have done till now, 10 steps of enzymatic activity, long steps, difficult names, and we have not achieved either water molecule or carbon dioxide molecule. So there must be something fishy or dicey in between this. So this step, which is the aerobic respiration step that takes place inside the mitochondria, would deal with the release of carbon dioxide molecules. Now we are going to see the Tca cycle or the Krebs cycle, where actually we are going to see how the pyruvic acid molecule, which had 3 carbons, which was a 3 carbon compound, is going to get rid of those 3 carbons in the form of carbon dioxide. It is all about getting rid of the carbon and taking up the energy whichever is stored in that form. Now when we talk about energy, we didn't get ATPs, we didn't get carbon dioxide. So all these steps are now going to take into consideration whatever was missing in glycolysis. So you see, we cannot undertake the entire process in one go, because then it would not give as much energy as we are able to trace. You know, when we slowly and steadily learn things, we are able to get more and again more experience out of it. Same way it is about this thing is also similar to that way only, that if we do it step by step, then we are going to achieve more energy outcome as compared to one go step, which would be nothing less than a chemical process where the glucose is breaking. It would be like burning a piece of charcoal. You would get black material over there. Over here you have to make sure that gas, carbon dioxide is released, water is released, and immense amount of energy from a single molecule of glucose has to be achieved. So that process would be considered in this cycle. That process is the breakdown of pyruvic acid molecule into 3 carbon dioxide molecules, and it would be a cyclic process. That means whatever product is being used, that has to be regenerated. So as you might have learnt about C3 cycle, that is Calvin cycle, that is the formation of glucose from carbon dioxide. In that is also a cyclic process. That means few substrates are involved and the outcome comes in our hand, and rest of the things are regenerated. Same way we need the outcome, that is carbon dioxide is to be coming into our hands, and the rest of the processes whichever were involved, including the substrates, they would be regenerated so that they can be used for the next cycle, that forthcoming cycle, whichever would take place further. Now let us see the fate of pyruvate molecule. Do remember there are 2 pyruvate molecules which are formed after glycolysis, and over here we are considering single molecule of pyruvic acid. The other one also gives the similar fate. It undergoes similar fate, that is it would also release the 3 carbon dioxides in the similar fashion that we are going to study now. Now pyruvic acid is a 3 carbon compound. It combines with coenzyme A, that is a biomolecule, that is a type of vitamin only, which is a form of modified vitamin, that is coenzyme A, and there is NAD positive in the proximity. Now what is this NAD positive? We did talk about it in the glycolysis process. NAD is nicotinamide adenine dinucleotide. Now what is its purpose? That it undergoes dehydrogenation. That means it is going to take up the hydrogen ions. So whenever I am saying dehydrogenation, that means some enzyme known as some sort of dehydrogenase. And some sort of dehydrogenase, when I say that if it is dehydrogenase, it is going to be of substrates dehydrogenase. So in the presence of enzyme pyruvate dehydrogenase, please remember this step. The enzyme is pyruvate dehydrogenase. Now as pyruvate is undergoing dehydrogenation, NAD positive would come there and take the advantage and get reduced to NADH and H positive. And at the same time, this pyruvate is going to get rid of one of its carbon dioxide, one carbon molecule. It would release one molecule of carbon dioxide and get converted to acetyl coenzyme A. Alright, now this acetyl coenzyme A is a 2 carbon compound. Now it enters the Krebs cycle. It is named after the scientist who discovered it, Krebs cycle. Tca cycle is the other name, and citric acid cycle is the third name for this cycle. All are same, nothing is different. These are the synonyms, you can say. The acetyl coenzyme enters the cycle and forms a 6-carbon compound. Now see the mathematics behind it. It is a 2-carbon compound and this is a 6-carbon compound. So by default, it would have combined with the 4-carbon compound. That is why we are having 6-carbon compounds. Now what is quite boring about these cycles is that you have to remember enzymes names as well. So the easiest way is that you find logic behind it. What is the enzyme doing? So in this process, as you can see, acetyl coenzyme has come and combined with water and oxaloacetic acid, which is a 4-carbon compound. We talk about this acid in C4 cycle, if you remember. Oxaloacetic acid, that is a 4-carbon compound, it combines with acetyl coenzyme A, that is a 2-carbon compound, and lead to synthesis of citric acid. Okay? Pay attention to the sentence again and pay attention to the word synthesis especially. Acetyl coenzyme A, oxaloacetic acid, which is 4-carbon compound, combines with a 2-carbon compound in the presence of water by taking a water molecule, gives rise or leads to the synthesis of citric acid in the presence of enzyme citrate synthetase. Alright, I said synthesis because synthesis is taking place, so the enzyme involved would be whosoever synthesis is taking place, that is citrate synthetase would give rise to citric acid. Now the citric acid in the presence of another enzyme, that is aconitase, undergoes 2 isomeric changes. One with loss of water and other with gain of water. It becomes cis-aconitic acid, which is again a 6-carbon compound. Nothing has changed. No carbon dioxide has been released or no carbon compound has been released. So it becomes cis-aconitic acid by loss of the water molecule. Here it takes up the water molecule. Please don't confuse yourself with uptake and release of water molecules because it is quite confusing, I believe it. But over here just cram it that here it is one water molecule that is being taken, here it is released, here it is taken back. We will consider those 6 water molecules in the end, but for now just remember that there are 2 places in the cycle where water molecules would be taken and in fact 3 places where water molecules would be taken and at one place it would be released. Now this isocitric acid and cis-aconitic acid, they are isomeric. They have been formed by the enzyme aconitase. Now this has nothing to do with logic, but because aconitic acid is being involved, so you can remember it from the term aconitase. Now when this isocitric acid is formed, what will happen? This 6-carbon compound, that is isocitric acid, it is formed only for one purpose, so that it can easily undergo the process of dehydrogenation. Earlier also I used the term dehydrogenation. Whenever I say dehydrogenation, that means an NADH would be formed. So you see, dehydrogenation takes place, NAD positive gets reduced to NADH and H positive, and there is a release of carbon dioxide. So this is the second carbon dioxide molecule that we got rid of. We were talking about 3-carbon compound, that was pyruvic acid. It got rid of one of the carbon molecules. It became acetyl coenzyme A, that was 2-carbon compound. Now we have got rid of another carbon in the form of carbon dioxide, and the enzyme which is responsible for bringing about this change, it is undergoing dehydrogenation. Okay, so whom it has dehydrogenated? It is isocitrate dehydrogenase. It is the third enzyme, that is isocitrate dehydrogenase. It got dehydrogenated, enzyme would be isocitrate dehydrogenase, which gives rise to alpha ketoglutaric acid. Alright. Now these are the 2 successive steps where we lose first carbon dioxide molecule and the second one, because the compound which had entered the cycle was a 2-carbon compound. So basically over here we have got rid of the carbon molecules, whichever carbon dioxide molecules, I beg your pardon, we have got rid of the carbon that we wanted to get rid of. So the next steps whichever would be formed, they would be for regeneration of oxaloacetic acid, so that another acetyl coenzyme comes and combines with it. So pay attention to this next step, that is alpha ketoglutaric acid would get converted to succinyl coenzyme A. Now as you see, this process is also of dehydrogenation. That means there would be alpha ketoglutarate dehydrogenase. That would be the enzyme which would be involved, which gives rise to one NADH and one CO2 molecule is released. What comes as an outcome is succinyl coenzyme A. This succinyl coenzyme A would undergo change to succinic acid in the presence of the enzyme succinyl coenzyme A synthetase. Okay. This enzyme, which is responsible for formation of succinic acid, is succinyl coenzyme A, or simply succinic synthetase, that you have to remember. Now, during this process, when this succinyl coenzyme A is getting converted to the carboxylic acid, what happens is a compound known as GDP along with inorganic phosphate forms GTP, which is coupled with another reaction where the ATP, whichever energy is being used, there is a coupled reaction. I need not to explain what coupled reactions are. ADP gets converted to ATP. Alright, so till now we have had 2 molecules of CO2 released inside the cycle. We are not considering this step, that is the first step. Inside the cycle we had 2 molecules of CO2 were released, 2 molecules of NADH were synthesized, and now during the regeneration we have got another molecule of ATP. Now what happens next is this succinic acid gets converted to fumaric acid in the next step under the activity of enzyme succinic dehydrogenase. Now again I am using the enzyme dehydrogenase. All these are 4 carbon compounds. Please remember, this is also 4 carbon, fumaric acid is also 4 carbon, malic acid is also 4 carbon. So this succinic acid under the activity of succinic dehydrogenase get converted to fumaric acid. Earlier when I used the term dehydrogenase, I had seen some sort of hydrogenation of NAD occurring. Now what would happen over here? Over here we'll have another reaction. Fumaric acid is involved. So we see that FAD positive, that is flavin adenine dinucleotide, getting converted to FADH2. Please remember this. Okay, over here when the dehydrogenation takes place, the hydrogen ions are picked up by FAD positive in this step. So we have another energy currency over here. FADH positive is being formed. Next, this fumaric acid takes up a water molecule and forms the malic acid. Again malic acid is being formed. So the enzyme involved is fumarase because it is breaking the fumaric acid into malic acid. Next step is malic acid is getting converted to oxaloacetic acid, and over here you can see again that there is formation of NADH. So by default the enzyme would be malate dehydrogenase. So this entire process has given us the 2 molecules of carbon dioxide that we wanted out of acetyl coenzyme A. At the same time it has regenerated the oxaloacetic acid, which started this entire cycle, and at the same time we were talking about the energy currencies. Those were also obtained. We do know that it is NADH ultimately which is going to give rise to the molecules of ATP. So we have those NADH as well. So what is the outcome? 2 molecules of pyruvate are there. So if you remember, what are we going to get after this Tca cycle? What was the outcome of glycolysis? Glycolysis had given us, for once let's talk about this. There is 1 molecule of NADH over here. Another 2, 3 and 4 molecules of NADH have been obtained. So we have 4Na. Alright, these are 4 molecules of NADH. At the same time, there is 1 molecule of ATP that is being formed with a coupled reaction of formation of GTP. Then we have 1 molecule of FADH2 that is being formed. This can be represented as NADH2 also. FADH is being formed. This is the complete outcome. And what has been released? 3 molecules of CO2 have been released, which would be gaseous. We don't know where it goes. It could be used anywhere. So 3 molecules of CO2 are being released, and this is the complete outcome that we obtained when 1 pyruvate breaks into, when 1 pyruvate gets rid of its carbon dioxide. So 1, 2, 3 and 4. Now see, this is 1 pyruvate, pyruvic acid. There is similar pyruvic acid which was formed out of glycolysis. So we are going to multiply this outcome by 2. So in total we are having, as a result of Krebs cycle, we are having 8 NADH molecules, 2 ATP molecules and 2 FADH molecules. This is the outcome of the Krebs cycle. Now add it with that of glycolysis. You remember how many of them were in glycolysis? There were 2 ATP which were generated, though there were 4 which were generated and 2 being used. So we get the total outcome to be 2, and there were 2 NADH molecules as well. So by the end of TCA cycle, what do we have in our hand is that we have 10 molecules of NADH and 4 molecules of ATP and 2 molecules of FADH2. And if you remember, in the beginning we had written that in the equation of respiration that there are 36 molecules of ATP which are being formed, and I see only 4 molecules. Now what we are going to study next is how these NADH and FADH are going to get converted into the molecules of ATP, because ultimate currency of the cell is ATP only, as you must be having lots of rupees and other currencies of your choice, but when you have to deal in international level you need dollars. Same is in the case of cells. You cannot use NADH or FADH2, you need ATPs basically. So those ATPs would be formed in the subsequent step, that of aerobic respiration is. Until now, remember, we have nowhere discussed oxygen. Now the next step, that is of the aerobic respiration, that is ETS, electron transport system. It would give rise to what was there in the equation, that is the oxygen was there and the ATPs. We would see that in electron transport system, which would take place inside mitochondria, not inside the not inside the stroma of or the matrix of mitochondria, but in the mitochondrial membranes. There are 2 membranes, you know that, mitochondrial membranes. So the mitochondrial membrane would act as the site for the formation of ATP, that is ATP synthesis would take place in mitochondria, and the usage of oxygen, when we say aerobic respiration is taking place, the usage of oxygen would be seen in ETS. Thank you.